Good afternoon. I am the pleasure to present Dr. Denis Servent, a great scientist. He is a head of the molecular engineer for health unity, head of the CNRS, toxin receptor and ion channels at the Université Paris-Saclay, Gif-sur-Yvette, France. About the education and qualification. He did his PhD thesis in biochemistry, Université Compiègne, France. Chemical Institute Natural Substance, and a postdoctoral position at the CNRS in Paris, France, with the X-ray crystallography laboratory in CNRS. In 2003, he was research direction habilitation in pharmacology at Paris-Descartes University, France. And between 1992 and 2000, he was research at the protein engineer department under the direction of Professor André Menes. Since July 2018, he's head of the molecular engineer for health unity. Other relevant information, he supervises 9 PhD students. He's member of the 32 theses in jury of other diverse theses. He is a teacher, professor at the National Institute of Nuclear Science and Techniques, a speaker in pharmacology in master's degree at the Museu Nacional of Natural History. And about Society of Toxinology, he's member of the French Society of Toxinology since 30 years, treasurer, and the president from 2008 to 2016. He's member of the International Society in Toxinology and invited the speaker, the National, European, and International Society of Toxinology, and member of other scientific organizations, had different grants, and participated in several conferences and meetings, including sometimes in Brazil. You are very welcome, Denis. So please, you can begin your presentation.
Hello, everybody. And first, thank you, Maria Elena, for this very kind introduction. And of course, to the organizing committee and Maria Elena in particular for inviting me. I apologize not being able to be with you in Brasilia. I'm sure that it will be a very nice meeting and an opportunity to discuss with all my colleagues and friends in Brazil. I hope that it will be the case for the next time. Anyway, I will try to present our research, which are focused in the last few years on GPCR interacting toxins, with the goal to develop interesting pharmacological tools or even for therapeutic applications. So, as all of you knows well, we are in face of more than 100,000 venomous species in animal kingdom, covering practically all the phyla, from cnidarian to insects, scorpions, spiders, reptiles, and even in mammals. And these venomous animals have developed a lot of molecules in their venoms in order to subdue their prey or to defend against predators. And these molecules represent approximately tens of millions of natural molecules if we are an approximation of 100 of different peptides in each venoms. And if we are looking at all the different species in snakes, scorpions, cone snail, and spiders, and these natural libraries of approximately 40 million of molecules, even if we are trying since 30 years now in the community of toxinologists to study these molecules, only a very small amount of this natural diversity have been discovered, have been described and studied. So we have the possibility to try to look more carefully on new interesting targets, how these natural molecules may interact and may be useful for potential therapeutic applications. Interestingly, even if the natural diversity of these molecules is very huge, in fact, peptides and proteins present in their venom, in these venoms, belongs to a quite restricted diversity in terms of structural scaffolds. Especially for small peptides present in these venoms, they belong to a quite limited number of structural family. Some of them are presented in these slides. For example, as you know, the ICK or notin scaffold, which are characterized by peptides between 20 to 35 amino acids with 3 disulfide bridges. And these peptides are highly present in spiders, in cone snails, and also in scorpions. And these peptides have been selected due to their high stability and their multipotent specificity, because they are known, for example, to be active on all the ion channels, sodium, calcium, potassium, ASIC or TRP channels. And also they can interact and block some specific receptors, such as the ryanodine receptor or the purinergic P2X3 receptors. The other scaffold is the cunit scaffold. This structure with longer peptides between 50 to 65 residues, 3 disulfide bridges. These type of peptides are present in spider, in snakes, in sea anemona, or in tick, and they are well known for their trypsin inhibitor property, for their ability to block potassium channels, and also, as we have shown in our lab more recently, to block the vasopressin B2 receptor. And finally, one of the last very common structural scaffold is the 3-finger scaffold, which is this time restricted to snakes and mainly to Elapidae snakes. These long peptides from 60 to 70 residue, including 4 or 5 disulfide bridges, are known from a long time now to interact with nicotinic acetylcholine receptors, but they are also active on ASIC or sodium channels, active on a lot of different GPCR receptors. They can block specific enzymes such as the acetylcholinesterase or PLA2, and they can also forming pore in the cell membrane to exert their cytotoxic property. So there are only few examples, but of course they are more large. And all these peptides exert their toxic effect by interacting on specific targets. For example, for neurotoxins, they can act on the central or peripheral nervous system, mainly by interacting on voltage-gated ion channel or on ligand-gated ionic channel, such as the nicotinic receptor, or, for example, also by blocking enzymes which play a crucial role in the control of the neuromediator concentration or release at the synaptic left, such as the acetylcholinesterase or the PLA2. Other molecules present in venoms are active on the hemostatic system and may block or activate the coagulation cascades, interacting, for example, on coagulation factors, on the fibrinogen fibrin, thrombin, prothrombin systems, on integrin receptor, etc. And finally, to be active on the cardiovascular system, peptides or enzymes present in venoms may interact specifically on ion channels present in heart. They can make pore on cell membrane of the cardiomyocytes or interact, for example, on specific enzymes such as the angiotensin converting enzyme. And quite surprisingly, in fact, among all these molecular targets, ion channel, receptors, enzymes, etc., in fact, not none of them, at the exception of the endothelin receptor, and we will come back later on this receptor, none of them are G protein coupled receptor. And it's quite surprising because G protein coupled receptor is the most important family, superfamily of receptor in metazoan. They are largely present in all the animal kingdom. They are involved in practically all the physiological processes. For example, in human, they represent 3% of the human genome. More than 800 different receptors belong to this superfamily. And in terms of therapeutic application, approximately 30% of all the drugs actually on the market are known to be active via their interaction with the GPCR. So it's the most important receptor target for drugs. As you know, GPCR are characterized at the structural level with this typical signature, including 7 transformable domains and in alpha helices structures. And despite this homology in terms of structures, this GPCR can be activated by a large difference of ligands, of agonists, that could be with light, for example, for the rhodopsin, that could be induced by calcium, by small molecules, odorants, pheromone, amino acids, nucleotide, small peptides, or even with very large proteins, for example, with the folliculin stimulating hormone, the interleukins, the chemokines, or the alpha-latrotoxins. So despite they are quite different size and they all interact on the GPCR. And according to the name of this family, the coupling and the activation of the agonist to this receptor induce the coupling of a small protein G here, which is a trimeric proteins with alpha, beta, and gamma part. And the coupling of this trimeric protein G following the activation induce the activation of different types of effectors, which could be enzyme, such as phospholipase C, for example, or adenylate cyclase, which are the most common, or some specific channels, potassium channels, for example. And the activation of these effectors induce the specific intracellular messenger release and activation of intracellular processes. More specifically, in the last 10 years, the pharmacology of GPCR was a little bit more complex, as I already present. In fact, one receptor activated by one agonist do not active only one signaling pathway. In fact, the same agonist can activate both what we call a G protein dependent pathway by this trimeric G protein, but also on another kind of activation and pathway related, for example, to the activation via the beta-arrestin proteins. That means that the same agonist, for example, in this case, the angiotensin 2, which activates the angiotensin 1 receptor, can simultaneously activate both G protein or dependent or G protein independent pathways. And that's interesting because some physiological function associated with these receptors can be activated separately by different kind of agonists. For example, here, these TRV agonists activate selectively only the beta-arrestin pathway, and that's useful, for example, to lead finally to cardio-beneficial effect via this receptor activation without any side effect which can be related to the G protein dependent pathway activation. And it's interesting for the pharmaceutical industries, which are now looking this specific kind of agonist. That's been agonist which can activate only one specific pathway and not all which are under the control of one receptor. That means that by this approach, we can hope to have more selective molecules without any side effect related to the more global activation of all the function under the control of one GPCR. In terms of family, the GPCR are classified in 5 different receptor subfamily. The most important one is the rhodopsin one, which includes more than 70% of all the GPCR. For example, in this family, we can recover the biogenic amine receptors, such as the adrenergic, muscarinic, histaminergic, serotoninergic receptors. We can find also the chemokine receptors. We can find all the olfactory receptors, and a large part of these receptors are still orphan receptors. We don't know exactly the agonist, the natural agonist of this superfamily. And so this rhodopsin family is clearly the most important. And after this one, you have also the adhesion family, the secretin family, the glutamate family, and the frisol taste family. These different class of GPCR are classified according to their structural homology. For example, the class A, the rhodopsin one, includes some specific residue here in red, which are conserved in all the GPCR which are included in this rhodopsin family. The class B, which is the secretin or GLP-1 receptor family, as you can see, there are a more important extracellular domain here with specific residue. So all the receptors with this characteristic are classified in this class B family. And finally, the class C are the family including the GABA A receptors, or the GABA B receptors, and also the calcium or the glutamate receptor. This family is characterized by a very, very important extracellular domain called the Venus flytrap domain, which serve to interact with these small molecules. And these family of receptors are mainly activated in the dimeric form. So, in terms of drug discovery, as I said, approximately 30% of all the drugs on the market are interacting on this superfamily. That represents approximately 900 different drugs, which are acting on more than 170 different GPCR. Here on the left, you can see all the GPCR at the periphery. The size of the circle is depending on the number of drugs actually on the market. For example, here for the histaminergic or dopaminergic receptor, approximately 50 different drugs are actually on the market or in drug developments for all the different dopamine receptor subtypes. That's the same for beta-adrenergic receptor or for muscarinic or serotonin receptor, for example. And similarly, you can see that GLP-1 receptors molecules or opioid receptors molecules are also highly present in these pharmacology. You can see here on the right that practically all the therapeutical areas are covered by molecules interacting on GPCR. That could include, for example, central nervous system disease, such as Alzheimer's disease, schizophrenia, Parkinson, or analgesic property or analgesic compounds, but also molecules for diabetes treatments, cardiovascular or hypertension activity, allergy, etc., obesity, etc., etc. In terms of pharmacology, these receptor molecules that interact on this receptor can be classified in different subfamilies. You have the full agonist, which can induce a complete activation of this receptor. Partial agonist, as related to their name, are activating only partially the receptor, antagonists which stabilize the receptor in their inactive states, and finally inverse agonists, that's a subclass of molecules which are able to block what we call the constitutive activity of this receptor. That means that a small proportion of this receptor can be in an activated state, even in the absence of agonist. And so this constitutive activity may have some interesting functional role. And so the molecules which are capable to block this constitutive activity are called inverse agonists. And we have also a large part of molecules interacting with GPCR, which are allosteric modulators. They can, in fact, modulate the interaction of the natural agonist by, in a positive way or in a negative way. It's what we call positive allosteric modulator. That means that the dose response activity of the agonist will be shifted on the left, or negative allosteric modulator decrease the affinity or the potency of the natural ligands. And again, these kind of molecules are the most interesting in terms of therapeutic application, because they are able to be very precise in their spatiotemporal control of the functional effect on this receptor. At the structural level, the two types of molecule can interact on two different sites. Agonist or antagonist molecule interact on what we call the orthosteric site here in red. For the class A receptor, this site is located in the beginning, in the top part of the transmembrane domain, while classically the allosteric molecules interact more extracellularly at the interface between the extracellular loops and the top of the transmembrane domains. That's the definition of allosteric compounds, which interact on the site outside the orthosteric site where the agonist or competitive antagonist interact. So, in terms of structure, a lot of new structure have been identified in the literature. I described in the literature since 15 years now, and of course it's mainly correlated with the new approaches such as the cryo-electron microscopy, and now, if I am not wrong, we are more than 1,000 different structures which have been accessible in the PDB. The first one, more than 10 years ago, was obtained by crystallographic approaches, for example, for the beta-2 adrenergic receptors. These structures were obtained by the group of Brian Kobilka, and were this kind of very interesting data were correlated with the Nobel Prize obtained by Brian Kobilka and Bob Lefkowitz for the pharmacological and structural knowledge of the beta-2 adrenergic receptors. So now we are able to know very precisely the structure of this receptor in different states, in the resting states, in complex with agonist and the trimeric protein G, such here for the beta-adrenergic receptor with this trimeric protein G. And we are able to know very precisely modification in the structure of the transmembrane domain, which are induced by the fixation of the agonist, and which are correlated with the activation of the signaling pathway following the agonist interaction. So we know a lot of structure covering all the different classes. For example, the class A, we have seen the beta-adrenergic structure just before. We know now, for example, how chemokine peptides interact on a dimeric form of the chemokine receptor, the CXCR4. We know for the class B how the GLP-1 peptides interact on the extracellular domain of the GLP-1 receptor, here in red, in green, with the trimeric protein G at the bottom, in the intracellular domain of the receptors. And we know the structure of class C GPCR, for example, here the GABA B receptor, the heterodimeric form of the GABA B, B2, B1B in complex, with this large extracellular part and this transmembrane domain we are linked by a linker. So for all this class of receptor, we have now very precise structural data, which are very useful for structure-based drug design, targeting specifically different receptor subtypes. So now, what we know about toxins which are interacting on this superfamily of receptors. Of course, if we compare the diversity of molecules which have been identified or characterized on this receptor, that's very, very few as compared with a lot of different molecules known to interact on ion channel, on nicotinic receptor, or on enzymes, for example. So probably less than 2 or 3 dozens of molecules have been identified in the last 20 years for interacting on GPCR. But of course, we discover regularly new molecules and with interesting property, as you will see in my presentation. We have classified these molecules in 2 different families. In the first one, the molecules are quite similar to the natural hormone or ligands known to activate this receptor. So these peptides present in venoms are in fact analog of the natural agonist of this receptor. That's the case, for example, for a lot of molecules known, identified and isolated from cone snail. For example, conopressin, which are analog of the vasopressin, conculacin G, which is an analog for the neurotensin, conorphamide, which are analog of R-famide peptide, or conorphine, which are analog to dynorphin. And also consomatin, which is analog from somatostatin. That's all for cone snail peptides. There are also molecules and peptides isolated from snakes, such as the MIT or prokineticin, which is an analog of the EG-VEGF factor, sarafotoxins, analog of endothelin, or exenatide from the Gila monster, which is an analog of the GLP-1. And we have the second family, which are non-agonist ligands. So the molecules are completely unrelated as natural agonists. And for example, that's the case of the latrotoxin, which interact on latrophilin receptor, toconotoxin on the somatostatin, the melanocortin on melanocortin receptor, angiotensin toxin for the angiotensin receptor, and all the different molecules identified from Dendroaspis snake, which interact on a large diversity of receptors. And so I will focus now on all these different examples in green, 3 from the agonist family, and this one from the antagonist family. The first one are sarafotoxins, and these molecules are the only one which are clearly related and correlated with the highly toxic effect of these toxins on the prey of Atractaspis, which is the only snake genus which are producing these molecules. These snakes, these mole viper, are located in sub-Saharan Africa, and they are capable to produce a very strong and very potent cardiotoxic and necrotic effect due to a strong vasoconstriction effect on the prey by acting specifically on endothelin receptor. And in fact, it's related to the very huge concentration of sarafotoxin in the venom of these Atractaspis snakes. And these sarafotoxin, as you can see here, are very close and very similar. The natural agonist of these endothelin receptor, the endothelin 1 and endothelin 2, There are small peptides of 21 residue known to activate the two different subtypes of endothelin receptor, endothelin A or endothelin B, which play a very important role in the control of the vasoconstriction or vasodilatation of smooth muscle cells or endothelial cells. And these two receptors are also important for pongogenic or mitogen activity in cancer cells. So the saraphotoxins are very similar in structure and possess all the classical residue in red, which are known to be important for the activation of endothelin receptor. Interestingly, more recently, new isoform of these saraphotoxin have been identified with a C-terminal extension here with 3 or 4 additional residue. And we have characterized some years ago that the addition of this extension produced a very drastic effect on the pharmacological property of this saraphotoxin. As you can see on this table, the natural endothelin 1 is a very highly potent molecule on both receptors, ATA and ATP, active from 20 to 150 picomolar affinity, with a quite small selectivity between the two receptors. The natural saraphotoxin is very potent, but more selectively on endothelin B, less potent on endothelin A. And as I said, the simple extension at the C-terminal part of saraphotoxin M or I3 completely abolished the activity on endothelin A and drastically reduced the potency for endothelin B. And when we cut and delete this extension in these two saraphotoxins, we restore a very potent affinity on ETB particularly. And for example, we were able to produce this saraphotoxin I3 CTR, which is in fact the most potent molecules actually known to interact on the endothelin B receptor, most potent and most selective ligands, interacting with 18 picomolar on ETB and only active at micromolar range on ETA. So it's a very interesting molecules to label or abolish selectively the ETB function in vivo. And this particular interaction is very coherent with the structural complex which have been obtained, both interacting with ET1 or saraphotoxin B in the endothelin B receptor. As you can see, both molecules are completely overlapping and interact at the top of the GPCR ETB receptor with this tryptophan 21, which play a very crucial role in this interaction and very critical role in the agonist property of these molecules, and explaining why when you add some additional residue at the C-terminal part of the molecule, you create, in fact, a very important steric hindrance effect, a steric clash, which conduct, which is correlated with the complete loss of the property of the interaction property of this molecule. This molecule could be very interesting at the pharmacology as pharmacological tools to selectively address the role of the ETB receptors. The second interesting molecules for agonist effect is the exenatides. These molecules have been, these molecules are in fact analog of GLP-1. GLP-1 is the glucagon-like peptide 1 molecules. As you can see here on the left, GLP-1 are small peptides which play a very important role in the glycemic control, because the GLP-1 may, in the pancreas, control the release of insulin or of the glucagon, and also by acting on a lot of different organs in liver, also in the brain or in the GI tract. The GLP-1 control, for example, a lot of different systems, particularly the satiety control. And GLP-1 analogs have been discovered in the 90s from the Gila monster, from the Eloderma suspectum saliva. And as you know probably, these molecules is in fact a very approach analog of the GLP-1. As you can see here, the molecule which was called initially exenatide or exendyne, but the same, is quite similar of GLP-1, but with specific modification, which in fact are very useful because GLP-1 molecules possesses a very short half-life in vivo, few minutes, only 1 or 2 minutes of activity in vivo, while the exenatide, due, for example, to this simple modification in the N-terminal part, the glycine insertion here, is sufficient to abolish the proteolysis activity of the DPP4, which hydrolyzes quite quickly the GLP-1, which is not the case for exenatide. And the C-terminal extension is also involved in the resistance of the carboxypeptidase by for the exenatide. That means that, for example, these molecules is characterized by a half-life in vivo of 1 or 2 hours, as compared to few minutes for GLP-1. So, of course, these molecules obtained the FDA approval in 2005 under the name of Byetta for the treatment of type 2 diabetes. And at that time, the mode of administration of exenatide was an injection 2 times per day of the molecule to have the maximum effect. And since that time, in 20 years, in fact, a lot of different analogs have been developed to increase the facility of the treatment and to decrease the frequency of administration. At the beginning, for example, a few years after the bidireon molecules derived from the initial exenatide with specific modification in the sequence was characterized by a longer time of activity, correlated with only one injection per day. After several other modifications, for example, the capacity of coupling albumin recognition site on the GLP-1 analogs produce a more stable molecule which are active 1 week. So the patient are injected only one time weekly, and that the most useful molecules actually used not only, of course, for the type 2 diabetes, but also for obesity. I'm sure that we have already heard about Ozempic or Wegovy molecules, which are all derived from the initial exenatide and which are actually injected 1 time weekly, or even the last galenic molecules are orally administered, that the Ribelsus molecule, which are orally available after encapsulation or specific oral delivery process for obesity or type 2 diabetes. So it's a real success story for naturally isolated molecule interacting on GLP-1 or GPCR. And finally, the last example of agonist molecule was discovered by the group of Elena Safavi and Mani in Utah University in US. Recently, Elena with Toto Oliveira have already characterized analog of insulin in some venous cone snail venoms, and to explain, in fact, you know that. I cut the sound. You know that, in fact, some classes of cone snail do not catch their prey by the classical strategy of the fish hunting strategy with harpoon, but they are able to, in fact, probably release in the proximity of the fish some very active molecules which are able to have a very strong effect on the fish. and the cone snail, as you can see on the movie, can swallow and absorb the fish without any activity or without any possibility for the fish to escape from the cone snail. That's the case, for example, for Conus geographus that you can see on the right. And so, of course, the main questions to explain this kind of activity is how to explain that the molecules can be released and have the capacity to paralyze or induce what Toto Oliveira called the nirvana cabal, to block the escape possibility of the fish. And to explain that, Elena and Toto proposed that molecules can be released and can provoke a very strong hypoglycemic effect on the fish. First, by releasing analog of insulin, the cone insulin molecules, and more recently, this effect can be reinforced by molecule identified by Elena called consomatine. And consomatine are analog of somatostatin peptides. As you can see here on the left, that's the natural hormone, somatostatin 14, which are known to activate the 5 subtypes of somatostatin receptor. And this consomatine peptide from this cone snail, it's in fact a very approach analog of this somatostatin receptor, and even more approach, in fact, of octreotide, which is a molecule which have been optimized by pharmaceutical industry and which are actually on the market for the treatment of different pathologies such as neuroendocrine tumors or acromegaly, for example. And quite surprisingly, molecules present in venoms possess specific modification, such as the presence of D-tryptophan in their sequence, which are exactly the same kind of molecule which have been optimized by pharmaceutical industry to optimize the property of this molecule to interact on this somatostatin receptor. So these molecules are capable to interact specifically on the somatostatin SST2, and this effect block, in fact, the release of the glucagon and potentialize the hypoglycemic shock that could explain the paralytic effect on the fish. So it's very interesting and very potent molecules, which are of course developed in the lab of Elena and Toto Oliveira for potential therapeutical applications, because these molecules are always more stable and more potent than their natural homologue. So as I said, in parallel to this agonist molecule, we are a lot of antagonist molecule. And recently in the lab, we have, thanks to a European program called Venomics program, we have produced quite large transcriptomic and proteomic libraries of near 200 of different venomous species. And we produce by recombinant expression or solid phase peptide synthesis approximately 300 different molecules, which can be screened on different receptors. And in this first study, we screen on melanocortin receptor, which are important receptor in different functions, for example, in immune function, in lipid metabolism, or in food intake control. And we discovered these two molecules from a spider or from scorpions with the ICK or CS alpha beta structure. And these molecules are able to activate the melanocortin 1 receptor or even to potentialize the activity of the natural agonist, the alpha MSH, which is quite interesting. But unfortunately, these two peptides are not selective at all for the melanocortin 1, 2, 3, or 5. And so this lack of selectivity preclude any other development of this molecule, which can be only interesting in terms of pharmacological activity, but not for therapeutical development. And that's the same case of these other peptides isolated from this conus lithoglyphus. And this peptide, again, is active in the 100 nanomolar range on melanocortin receptor. After structure activity relationship studies, we were able to optimize a little bit the selectivity, but not for MC4, which is the most interesting melanocortin receptor to be addressed, but unfortunately not sufficiently to go further in the development of this kind of antagonists. More recently, Anne-Cécile in our lab during his PhD identified also from this conus melioraris an interesting peptide active on angiotensin receptor, that the first toxins present in venoms known to be active on this family of receptor. Again, this molecule is active on angiotensin 1 and angiotensin 2, sorry. As you can see, the interaction of the fluorescent angiotensin is abolished with an excess of these peptides. Peptide, which is a full antagonist on the different pathway, the GQ pathway producing the inositol phosphate release, which is completely abolished in the presence of the toxin, or also the MAP kinase pathway. You can see here the ERK phosphorylation of ERK, which is abolished in the presence of the toxin. And interestingly, this toxin is at 100 micromolar is not able to activate or block other receptors which are active on the cardiovascular system, such as the endothelin, phelin, or vasopressin receptor. But again, here, the quite limited affinity in the just sub-micromolar range is not sufficient to go further and to develop this molecule as potential therapeutic application. In fact, the older molecules known to be active in venoms and interacting on GPCR have been discovered by two groups in the 90s, by the group of Evert Carlson and by the group of Lincoln Potter in the 1992, in the 90s. And these molecules have been discovered in the venom of Dendrospis angusticeps or Dendrospis polylepis. And as you can see, these molecules are 3'4" peptides, and they are quite active on muscarinic receptor with quite different kind of affinity and selectivity. For example, as you can see here, the MT7 peptides is a very potent molecule peptides active on M1 receptor in the picomolar range, and this MT7 is not active at all, even in the micromolar range for all the other muscarinic subtypes. So it's the most potent and selective antagonist of the muscarinic M1 receptor. So in the past, we have characterized a lot of different molecules from these venoms on different kind of receptor. Here you can see, for example, the screening of all these molecules, not the screening, the pharmacological characterization of these different kind of peptides on 9 different receptors, 2 from the muscarinic receptor, N1 and 4, 3 alpha-1, 3 alpha-2 adrenergic receptor, and the dopamine 3 receptor. And as you can see, the different toxins present in these venoms may have completely different pharmacological profile. Some are totally selective and highly potent for only one receptor, MT7 for M1, MT alpha for alpha-2 adrenergic, alpha-2B adrenergic receptor, totally selectively, while, for example, the MT3 toxin is a non-selective toxin active on practically all these receptors, muscarinic, adrenergic, dopaminergic, etc., in the nanomolar, in the 10 to 100 nanomolar range. And of course, one interesting question is how these molecules can interact so differently and which could be the differences in their sequence, because they are all 3'4" toxins, which could be correlated with these different kind of pharmacological profile. It's a very tricky question, and we have not yet the answer. But we are able by shuffling the loop strategy to be able to characterize different regions of this molecule which are associated with their selectivity. For example, we are able to create by switching this region of the toxin MT1 onto the MT7 toxins, we create a chimeric toxin which are not only active on M1 as the initial one, but also active on M4. So we are now able to identify some regions which are associated with some specificity of interaction with different receptor subtypes. In terms of adrenergic toxins, we have identified, for example, these toxins selective for the alpha-adrenergic 1A. This receptor is very important in the prostate function, and we have shown that these molecules is active on human prostate biopsy and abolish the activation of the norepinephrine on this receptor. And this activity is also important for abolishing the pressure in the urethral arteries and not in global general arteries, which is quite interesting for developing drugs for treating benign prostatic hypertrophy. But some years after this discovery, a small molecule called tamsulosin, specific of the alpha-1A receptor, reached the market. And so we cannot compete with a quite large peptide in competition with a very small molecule with the same kind of properties. Interestingly, when we compare the mode of action of these different molecules, they even if they are in the same structural family, the 3'4" family, they interact on the same class of GPCR, muscarinic M1 or alpha-adrenergic alpha-1A. They interact completely differently on these two types of receptor, and they interact on different regions of the receptor, the extracellular loop for the muscarinic receptor, or residue more in the orthosteric site for the adrenergic toxin. And the structure of the complex of the MT7 on the M1 receptor was solved by the group of Kobilka 4 years ago, and confirming the structure activity studies in our lab and mutational studies with the tip of the loop 2 and the finger 1, which interact with the extracellular domain of the M1 receptor at the origin of the selectivity and allosteric property of this toxin. And more recently, by a screening approach, we have discovered also a new molecule, the MT9, again from the black mamba. This molecule is quite atypical in the 3'4" family. They are shorter, with a very short loop 2 in cyan here as compared to the MT7 in green. And this molecule interacts selectively on the M2 muscarinic receptor subtype, as shown here. And it is not active on M1, M3, M4, or M5. And M2 muscarinic receptor is highly located in the heart or in the cardiovascular system, and is important in the control of the heart and the arteries via the parasympathetic system. And we have shown that this MT9 peptides is able to induce the relaxation of rat mesenteric arteries and also in human mammary arteries. And so we are still developing with our colleague in Nantes to see if this molecule could be useful in some pathology involving specifically the M2 receptors. To conclude, I would like to focus on one molecule which has been discovered by Nicolas Gilles in the lab more than 10 years ago now, the mamba quercetin, called MQ, which is a highly selective ligand of the V2R receptor, again isolated from the Dendroaspis snakes, with highly potent activity in the nanomolar range, not active at all on the other receptor on the same family, the vasopressin V1, V1A, V1B, the oxytocin receptor, or even not active on more than 130 different targets at 1 micromolar concentration. So it's very highly selective and potent ligands. And it's a fuel antagonist on all the signaling pathway, cyclic AMP pathway, but also beta-arrestin or MAP kinase pathway under the control of the V2 activation. Interestingly, this molecule is a Kunitz-fold, as you can see, and interact with the tip of the loop here, because when you mutated residue in this part, such as here in blue, you decrease a lot the affinity of these molecules. And as I said previously, this fold was known to be very potent for trypsin inhibition. The natural toxin is not active at all or only at the 10 micromolar range, so very low affinity. And when we transfer these two residues, which are important for the trypsin activity, we increase a lot the trypsic activity on this membrane scaffold. And the same for potassium channel inhibition. Jan Tilgat in Leuven University characterized this interaction. The natural toxin is active only in the 10 micromolar range. And when we graft at the N-terminal part this lysine residue, which is crucial for this activity, we increase selectively this activity. And this perfectly understanding with the recent POEM structure of the complex of the membrane with the V2R receptor, which have been solved by our colleague in the EGF in Montpellier. As you can see, the membrane is interacting at the top of the V2R receptor with the tip of the bottom of the structure interacting more closely with the extracellular domain or the first part of the orthosteric site of the V2R receptor. When we focus on this region, we show that this tip of the residue here, and particularly the two aromatic residue, phenylalanine 16 and 17, are highly important and critical for this interaction, and mimic other ligands such as the tolvaptan, which are also antagonists of this V2R receptor. And interestingly, membrane can partially structurally overlap the natural agonist AVP, the vasopressin, in cyan here. We can partially overlapping these two molecules, explaining the capacity of the MQ toxin to interact and to block the vasopressin V2 receptor. This receptor is highly selective and only located in the kidney and play a very, very important role in the water homeostasis and in the water reabsorption in the nephron, and is particularly located in the principal cell of the collecting duct of the kidney. And activation of the vasopressin on this V2A receptor is the main factor for controlling and is correlated with the antidiuretic effect. But suractivation of this receptor could be also involved in the activation of the MAP kinase and the proliferation of the kidney cells and of cysts, which can be correlated with the appreciation, with the decrease of the kidney function and correlated with some pathology, such as the polycystic kidney disease or to hyponatremia, which are clearly correlated with the surexpression and suractivation of the V2R receptor. So what was done by Nicolas, it's first to observe the in vivo effect of the membrane. And as you can see here, with the dose response effect create a diuretic effect in vivo, which is a clear effect of the antagonist property of this membrane on the V2R. And we are able to synthesize and to label this molecule with PET imaging molecules with the 18-fluorophore, which can be visualized by positron emission tomography. And as you can see, this membrane with these F18 labeled probes is highly located and totally selectively located in the kidney. And in pathological model in rat, first for hyponatremia, we show that the mambaquatin administration can restore the hyponatremia induced by the DDIVP as compared to the tolvaptan, which is the molecule which was reached the market 2 years ago, but more potently. And in the other model of a disease correlated with the V2R, the polycystic kidney disease, which is correlated with the increasing in size of the kidney and the lot of cysts in the kidney, which decrease the efficacy of the kidney function. We show that administration of the toxin in vivo reduce the size of the kidney and restore the kidney function. And these properties are really interesting and support the creation of the startup called V4Q, which was created this year in 2024, and to develop the potential therapeutic potential of the mambaquatin in this context of the kidney disease. So finally, I would just like to mention that peptides interacting with GPCR or even for ionic channels could be also interesting for imaging agents in cancer, due to the fact that a lot of these receptors or channels are surexpressed in different kinds of cancers, and we can address and develop some peptides with specific props to be able, by PET imaging, to image and to have a control of the development of the disease and to the efficacy of the treatment using this molecule as imaging agents. So to finish, I would like, of course, to thank all my colleagues of the toxin receptor and HLL laboratory, and in particular Nicolas Gilles, Pascal Kessler, Jordi Mago, and all the other colleagues which are involved in all this work. All my collaborators which are involved in the different projects presented during this presentation, the support which permits the development of all these projects. And of course, I would like to thank you, all of you, for your attention. And I'm ready to answer to your questions.
Dr. Denis, is very dense and very interesting, very big, your presentation, very rich.
Thank you.
And I think you have the subject for all the domains here. So I hope the people have interesting questions, because it's so much rich, your research and your presentation. Congratulations. A question? Rui, please.
Hi, Denis. How are you?
Hi, Rui.
Good
Nice
to
to
see
meet you. Nice to see you again.
Denis, could you talk a little bit for us about the deliveries of these molecules? How can the approach for delivery to the cells?
No, it's an important.
Excuse me, perhaps I will complete not only the delivery, but the stability, because most of these molecular, the peptides, do you think about it?
Yeah, you're right. It's a very important questions, and because the potency or the efficacy of the molecule is important, but of course the pharmacokinetic and the stability of the molecule in vivo, it's also very important. And so we can first, of course, we try to address mainly pathologies which are not related to receptors which are present in the central nervous system to avoid any problem with the crossing the blood-brain barrier. Because even if it's possible to address these questions by some specific addressing process, it's quite difficult. So we focus mainly on pathologies related to receptors which are more easily located in peripheral organs, such as the kidney, such as the heart, for example. But again, it's important to increase the stability. When we characterize, for example, the half-life of mamba-quercetin in vivo, it's quite short. In fact, it's 30 minutes. So it's probably too short to produce a very prolonged effect. So we have different ways to manage to address this question. The first one, and we are trying to do that, to use classical process which have been already used for peptide active molecules. That means for coupling these peptides with a specific sequence to be coupled to albumin, for example, which increase a lot the stability of the molecule in vivo, or to coupling to FcRn molecules to increase its half-life. Or we can also develop, and Nicolas do actually this kind of experiments to, for example, for hyponatremia, we need only very acute treatment. So IV injection could be managed mainly because hyponatremia are very often pathologies which are treated at the hospital. So we can manage quite easily some punctual IV injection. But specifically for polycystic kidney disease, which is long-term pathology, we need repetitive administration. And in this case, we are developing some specific pump and we have already used specific mini pump to deliver continuously in mice or rats models the molecules and to prolong the mode of the activity. Of course, is not is a little bit. It's still actually developed. For example, for small molecules, you have a small osmotic pump in human, which have been developed for delivering peptides. So we can do that, or we can try to optimize the in vivo property of the molecule to stabilize and prolong its effect.
Okay, more question? Denis, some time ago I heard a talk of you about you are searching for a toxin, I think it's from a snake, that blocks the receptor for control the bowel. It was finished. I think you presented here in the first International Congress in Brazil. That the connection is not good, excuse me. In a blocker for muscarinic receptor to control to control the empty of bowel.
Okay. I'm not. Sorry, I have not this molecule in mind. We have these adrenergic toxins which are active on adrenergic receptor, but it's for prostate as I presented quite rapidly. These molecules are active on alpha adrenergic receptor and important for prostate function.
Okay, you presented now. Okay.
It's probably this one. And as I said, we stopped, in fact, the development of this molecule because a small molecule called tamsulosin, which reach the market for exactly the same pathologies. So we decided to stop the study on these peptides.
Okay. And I know that you have a big library of conus toxins. And my question is, these conotoxins are very similar between the different species, or you have different constitutions in these venoms? Because you know that you have all in family of a different target for these toxins, no?
Yeah, yeah, it's probably, you're right, the conotoxins is probably the most important and diverse libraries of toxins, targeting the most larger diversity of targets. And we collaborate with different teams which are involved in the collection and in the milking of cone snails. We have not this expertise in our lab, but we are collaborating, for example, with Sébastien Duterte in Montpellier or with other people. And as you said, effectively, there are really important difference in the composition of the venoms of these cone snails according to their different locations, and also according to their mode of if there are piscivorous, vermivorous, or malacophage. That impact a lot the composition of the venoms.
And
as you know,
all these molecules are the peptides, or did you find some different molecules? All the peptides are all the peptides.
There are also small molecules which are not peptides in cone snails, but the main part of cone snail venoms are peptides, from very, very small peptides without any disulfide bridges to larger peptides, 30, 40, 50 residues with 3 or 4 disulfide bridges. But there are quite large diversity of molecules with very large diversity of targets.
Okay. Okay, if you have one question. It's
Hi, Professor Denis. Sorry to bothering you with a boring question.
No problem.
In the initial slides, you were speaking about the molecular target of the toxins, and you mentioned AC2, right?
Yeah.
And so I think this is the same enzyme that is used by several viruses, including coronaviruses, to attach and start pathogenesis. So this could be potentially used in antiviral therapeutics.
In fact, is not exactly the same. It's in the case of coronavirus or other virus, you're right, it's ACE2. But for the peptides, targeting angiotensin converting enzyme, is ACE1, not ACE2. Is not the same. And as you know, the main part of this research was from a Brazilian team, perhaps 50 years ago. It was the origin of the captopril, which was developed for the treatment of hypertension, and which was initially identified by Professor Rocha Silva, if I am not wrong, in Brazil, and it was from
It
was from sorry,
yes.
And it was from the BPP, bradykinin potential peptide, which have been after that modified to create the captopril. But it's interacting on ACE1 and not on ACE2.
And the seraphotoxins, I found it very interesting. But do you think it will be useful for the clinical? The point of view from clinical.
It's an interesting point. We have tried to do that and to have some interesting development in this point, particularly for cancer treatment. As I said, endothelin receptors are overexpressed in cancer and are involved in the metastasis process of glioblastoma or melanoma cancers, where these receptors are overexpressed. And there are people which are actually developing, for example, monoclonal antibodies specific for ATP receptor. So some of these toxins presented could be interesting, but in fact, we need to be the most potent as possible. We need to select specific subtypes of receptor in terms of structural. I'm not sure to be clear. In fact, we need to stabilize the receptor in a specific conformation, and the toxin, as far as I know, is not able to stabilize the receptor in the correct structure. In fact, the sarcotoxins are agonists, and we need, in fact, for this therapeutic effect, to have antagonist molecules. It's why the people are developing monoclonal antibodies with antagonist property for ETB. So if we success to change the property of sarcotoxin and create some very potent antagonist molecule, that could be interesting.
Mm-hmm. Very nice. Thank you. Thank you so much for all the time accept our invitation, and you hope
thanks
that
again, Maria Elena. And
yeah,
I've a nice meeting. Thanks you.
Thank you very much, and you hope in the next meeting you can be here. Thank you, Denis.
Thank you. Bye.
Bye.
Bye. Bye.